All right, guys. Today we're going to discuss nucleic acids. If you remember at the beginning of the year, we talked about big macromolecules. We talked about carbohydrates, we talked about lipids, we talked about proteins, and we also talked about nucleic acids. This is a really good transition unit between what we discussed in mitosis and meiosis and what we're going to discuss after this, which is genetics. And so if you have a good understanding of the structure and the function of nucleic acids, you'll have a much better understanding of what's to come in genetics. I'm going to start off by showing you a short video of the history and discovery of DNA, because there were a lot of scientists that played a role in its discovery. Ultimately, Francis Crick and James Watson got credit for discovering the structure of DNA, which is one of our main types of nucleic acids, but there were other scientists that played very important roles in its discovery. So enjoy the video.
We are in 1953 in Cambridge, a city where scientists are racking their brains to corner what was known as
We have discovered the molecule of life!
Yes, that one. Okay, let's rewind a little. Several researchers over many years discovered that in the nucleus of our cells there was a substance called DNA, which contained all our genetic information, and it was made of 4 compounds: adenine, thymine, cytosine, and guanine. How was it possible that the whole book of our life was written with just 4 letters? Our celebrating friends James Watson and Francis Crick thought that maybe the answer was in the structure of DNA, and they were trying to solve this 4-pieced puzzle that was driving all the scientific community crazy. They had 2 clues. The first one was a discovery made by Erwin Chargaff, who found that in all organisms he tested, the relative amounts of adenine and thymine were equal, and the same happened with guanine and cytosine. The second clue came from Rosalind Franklin, an experienced chemist who generated DNA crystals and shot X-rays through them to study their structure. The X-ray picture showed a repeated pattern, which meant it was something similar to a helix. Watson and Crick tried all the different possible unions between A and T and G and C, and one was perfect. 2 bonds between A and T and 3 bonds between C and G would fit in a helix. A double helix indeed. Also, the 4 units structured this way could code a huge amount of data, as a dot and a dash can compose a whole alphabet in Morse code. On top of these discoveries, specific pairing suggested the copying mechanism for DNA, for the production of new cells and new life. 10 years after the celebration in the pub, Watson, Crick and Maurice Wilkins, the boss of Franklin, received the Nobel Prize for this discovery, and the DNA revolution spread across every biology laboratory in the world.
As you saw in the video, Chargaff and Rosalind Franklin had a big role in discovering the function and the structure of DNA. It talked about the base pairing rules, which we'll discuss a little bit more, but adenine and thymine and cytosine and guanine. And those are Chargaff's rules. He said that A, which is adenine, can only bind with T, which is thymine, and C, which is cytosine, can only bind with G, which is guanine. And then Rosalind Franklin was really instrumental in understanding what the structure looked like through X-ray crystallography, which is just a really fancy way of saying she took a picture of the DNA molecule using X-rays that allowed Francis Crick and James Watson to actually discover this beautiful double helix structure of the DNA molecule. Okay, so here we're going to talk just about the definition, basic definition of what a nucleic acid is. And it is a macromolecule, so one of those really large molecules that we talked about at the beginning of the year, and it contains the genetic information in the form of genes. And so genes are small sections of the DNA that will actually code for a functional protein. Not all of your DNA will code for protein. Actually, a relatively small amount of your DNA codes for protein, but those sections that do are what determines your traits. It determines whether you have freckles or long fingers or a big nose or big ears or what color skin you'll have or hair or eyes. So all of that comes from the proteins that are being made from your DNA. There are two main types of DNA, and two main types of nucleic acids. And the first one we're going to discuss at length is DNA. And we'll talk about what DNA stands for in the next slide. DNA is what we typically think of as what we pass on from parent to offspring. We just discussed this in meiosis. So the passing on of chromosomes. So we're talking about DNA there, taking half of our instructions from mom, half of our instructions from dad. The egg is fertilized by the sperm, so now we have 46 chromosomes if we're talking about a human, and those chromosomes contain all of the instructions for making you. RNA is kind of like DNA's helper. RNA will become really important in taking those instructions from DNA and actually turning them into a functional protein when we talk about protein synthesis. So DNA stands for deoxyribonucleic acid. I want you to practice saying that at home, practice in front of a mirror so that you can get really good and you can tell your parents, hey, we learned about deoxyribonucleic acid today. And then they'll just be like, wow, you're so smart and you have a wonderful biology teacher. I know we tend to say DNA, but I want you to make sure you know what it stands for, partly because of this section here. So it tells you it's a nucleic acid, so we know what type of macromolecule it is. But the deoxyribo part also will tell us what type of sugar is making up the backbone of our molecule. And this will change when we talk about RNA. DNA holds the code for building proteins from amino acids, and this will make a little more sense when we talk about protein synthesis and how we actually use the instructions that are encoded in the DNA molecules to actually make proteins. That gives us the characteristics that we have. There are 3 critical things that genes must do. And genes are just small sections, again, on the DNA that code for a particular protein. So it's like a cookbook. And if you complete this step of the cookbook, you'll make a cute little cupcake, and it's something that you can eat. The instructions in DNA will help our body make proteins. So one of the critical things that a gene must do is that it must be able to carry information from one generation to the next. We received all of the DNA that we have from our parents. 23 chromosomes from dad, 23 chromosomes from mom. If we have offspring, we also need to be able to give them a set of DNA. If we couldn't do that, then the cell would not live. It wouldn't have the proteins that it needed to function. Genes also carry the code to make a protein. So it's not just carrying instructions, but our cells need to be able to read those instructions and actually make something useful from them. And those useful items are going to be proteins. And lastly, which is what we'll talk about next class, is that they must be copied easily. So we know that in interphase, before mitosis and before meiosis, we have to replicate all of our DNA so that the daughter cells have a copy of the DNA. If it's in mitosis, we want them to end up with 46 chromosomes. If it's in meiosis, we want them to end up with 23 chromosomes. But those chromosomes initially have to be replicated, and we need an easy and quick way to do that. And luckily, the structure of DNA lends itself to being copied very easily.
You have about 20,000 genes in your DNA. They encode the molecules that make up your body, from the keratin in your toenails, to the collagen at the tip of your nose, to the dopamine surging around inside your brain. Other species have genes of their own. A spider has genes for spider silk. An oak tree has genes for chlorophyll, which turns sunlight into wood. So where did all those genes come from? It depends on the gene. Scientists suspect that life started on Earth about 4 billion years ago. The early life forms were primitive microbes, with a basic set of genes for the basic tasks required to stay alive. They passed down those basic genes to their offspring through billions of generations. Some of them still do the same jobs in our cells today, like copying DNA. But none of those microbes had genes for spider silk or dopamine. There are a lot more genes on Earth today than there were back then. It turns out that a lot of those extra genes were born from mistakes. Each time a cell divides, it makes new copies of its DNA. Sometimes it accidentally copies the same stretch of DNA twice. In the process, it may make an extra copy of one of its genes. At first, the extra gene works the same as the original one. But over the generations, it may pick up new mutations. Those mutations may change how the new gene works, and that new gene may duplicate again. A surprising number of our mutated genes emerged more recently, many in just the past few million years. The youngest evolved after our own species broke off from our cousins, the apes. While it may take over 1 million years for a single gene to give rise to a whole family of genes, scientists are finding that once the new genes evolve, they can quickly take on essential functions. For example, we have hundreds of genes for the proteins in our noses that grab odor molecules. The mutations let them grab different molecules, giving us the power to perceive trillions of different smells. Sometimes mutations have a bigger effect on new copies of genes. They may cause a gene to make its protein in a different organ, or at a different time of life. Or the protein may start doing a different job altogether. In snakes, for example, there's a gene that makes a protein for killing bacteria. Long ago, the gene duplicated, and the new copy mutated. That mutation changed the signal in the gene about where it should make its protein. Instead of becoming active in the snake's pancreas, it started making this bacteria-killing protein in the snake's mouth. So when the snake bit its prey, this enzyme got into the animal's wound. And when this protein proved to have a harmful effect and helped the snake catch more prey, it became favored. So now what was a gene in the pancreas makes a venom in the mouth that kills the snake's prey. And there are even more incredible ways to make a new gene. The DNA of animals and plants and other species contain huge stretches without any protein-coding genes. As far as scientists can tell, it's mostly random sequences of genetic gibberish that serve no function. These stretches of DNA sometimes mutate, just like genes do. Sometimes those mutations turn the DNA into a place where a cell can start reading it. Suddenly, the cell is making a new protein. At first, the protein may be useless or even harmful, but more mutations can change the shape of the protein. The protein may start doing something useful, something that makes an organism healthier, stronger, better able to reproduce. Scientists have found these new genes at work in many parts of animal bodies. So our 20,000 genes have many origins, from the origin of life to new genes still coming into existence from scratch. As long as life is here on Earth, it will be making new genes.
Okay, so now that you know a little bit more about what nucleic acids are, what DNA stands for, we want to talk about where we can find DNA. So if we're talking about a prokaryotic organism, prokaryotes are things like bacteria. They do not have a nucleus, so that's this guy on this side. The nucleus is not there. It's not found in these types of organisms. So pro means before, karyon means nucleus, before there was a nucleus. So the DNA is located in the cytoplasm and usually in a single circular chromosome, sometimes called a plasmid. These are usually looped together, and it's kind of just floating around inside the cell. We are not prokaryotes. I apologize. We're not prokaryotes. We are considered eukaryotic organisms. And what that means is that we actually do have a nucleus in our cells, and our DNA is located inside of the nucleus in the form of linear chromosomes. It's not in one long piece of circular DNA. It's in 23 pairs of chromosomes, or 46 chromosomes total. We actually have about 6 feet of DNA in every single cell of our body. And so how does 6 feet of DNA fit in your cell, right? You can't even see one single cell with the naked eye. You have to look at it under the microscope. So how do we package 6 feet of it inside of these tiny, tiny, tiny cells?
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. The nucleosomes are packaged into a thread known as chromatin. This fiber is then looped and further packaged using other proteins which are not shown here. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated. At other times, as we can see now after the cell has divided, our DNA is less highly organized. It is still wrapped up around the histones, but not coiled into chromosomes.
Okay, so let's look a little more specifically at the structure of DNA itself, so our deoxyribonucleic acid. And some of this you may already know, but the shape of DNA is called a double helix. It kind of looks like a twisted ladder or a spiral staircase. So there are kind of like these outside, there's an outside backbone. So if you think about a ladder, like the 1x2s that are on the outside of the ladder, if we're talking about an old wooden ladder, and then the rungs in the middle will connect both sides of the ladders together. So the sides are made up of a sugar phosphate backbone. And in the center, holding the sides together, are base pairs held together by weak hydrogen bonds. We'll talk more about why they need to be weak when we talk about replication. We want to make sure that both sides of our molecule stick together because we want to take care of our DNA. We want to protect our DNA. We need to ensure that it's safe and that there aren't any mutations that are occurring, but we also need a way to copy it. And these weak hydrogen bonds are going to be key in how we can copy our DNA in order to replicate our cells. And the shape, like I said at the beginning, was first discovered by Francis Crick and James Watson, who are pictured here. I know this is a video clip, but we'll watch it at another time for the sake of time. Here's a really good few images of what the DNA structure looks like. So when I talk about a twisted ladder, that's this image kind of here. So you can see that here's one side of the ladder, here's the other side of the ladder, and in the middle we have these rungs that are attaching or keeping both sides together. And it is helical, so it is twisted. If we look at it a little more closely, what you see is that there's this phosphate group here, and then this shape here that looks like a little house. That is the actual sugar molecule. So when we talk about the sides of the DNA molecule, it's made up of sugars and also phosphate groups, sugars and phosphate groups. And right in the middle, these are our base pairs. They're nitrogenous base pairs. And when we talked about or heard about Chargaff's rules at the beginning, in the very, very first video that kind of gave us a history, this is what he helped us figure out, is that the A base is always paired with the T base, and G is always going to be paired with C, and holding them together are these weak hydrogen bonds. So A's and T's go together, C's and G's go together. And there are only 4 nitrogenous bases, adenine, thymine, cytosine, or guanine, in our DNA molecule. Seems really simplistic, but the order and the sequence of those bases is what allows us to make all of these different types of proteins. So our nucleic acids are made up of monomers. So remember, monomers are kind of like the small building blocks. They're like one Lego, in a tower of Legos. So our building block for nucleic acids are called nucleotides. And you can see the basic structure here. So we have some type of phosphate group, we have a sugar, and then we have a nitrogenous base, and those bases can change. Nucleotides are the building blocks of DNA, and hopefully you remember that term when we talked about macromolecules at the beginning of the year. So nucleotides are the monomers, nucleic acids are the polymers. The three parts again are the phosphate in yellow, and so you can see it in the simplified diagram at the top, which is something that you'll be drawing. And then you can also see it in this diagram at the bottom, which is a little bit more complex. In DNA, remember the letters stand for deoxyribonucleic acid, and so our sugar is a 5-carbon deoxyribose sugar. And then right in the middle, we have our nitrogenous bases, and if you remember in the last image, those bases are pointing towards the center of our DNA molecule. They're the rungs of our ladder. And so just to kind of stress this a little bit more, because this is a really important idea, is that the two sides of our ladder, okay, remember, which are made up of sugar and phosphate, are held together with our nitrogenous bases and our weak hydrogen bonds. And we follow base pairing rules that Chargaff actually figured out in his investigations. So our 4 possible bases, you'll learn to say A, T, C, and G, but I want you to also recognize their names. So adenine, thymine, cytosine, and guanine. Those are some more terms that you should be practicing in the mirror. So you can tell your mom, hey, DNA is made up of nitrogenous bases. Those nitrogenous bases are adenine, thymine, cytosine, and guanine. And they're going to be like, what? You're amazing. Even though you already are, this just makes you even more amazing. But the bases are going to be held together with weak hydrogen bonds. They need to be weak so that when we open up the molecule to make copies of it, we can do that fairly simply. And the DNA bases pair up in a complementary way. What that means is that we have pairs that will only pair together. So adenine will only pair with thymine, cytosine will only pair with guanine, and it's because of their chemical structure, which I will show you in just a second. But get that in your head. If we're talking about DNA, A goes with T, C goes with G, and that is what's forming the inside of our double helix. So here's a little bit better picture of what's going on. So you kind of have to turn your head sideways. But here are the little blue structures. Those are our phosphate groups. And this is our deoxyribose sugar. So the blue and the white, or the gray kind of, this section, this area, makes up your sugar phosphate backbone. And then the bases, our nitrogenous bases, would be pointing towards the center of our DNA molecule. So you have 2 categories of them. So there are purines. Our purines are adenine and guanine, and they have these 2 ring structures that form the nitrogenous base. Our pyrimidines are cytosine and thymine, and you can see that they're only a 1 ring structure. And so when our bases pair up, remember A goes with T, and C goes with G. So you have 1 purine pairing up with 1 pyrimidine. We can look at it a little bit further. So this is a little more close up. But on the outside, we have our phosphates and our sugars. In this case, deoxyribose sugar, because we're talking about DNA. And in the middle of our double helix, you can see that we're following our base pairs. So A with T, C with G. So the base pairing rules become really important when we need to replicate our DNA. So before mitosis, before meiosis, so in interphase, we have to replicate all of our DNA, and we need a really quick way to do that. And if we follow our base pairing rules, by knowing one side of the DNA molecule, we can really easily fill in the other side. And so this video is going to show you how that works, and we'll actually practice this in class.
DNA contains two strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes three parts: a phosphate group, a sugar molecule, and one of four bases: adenine, guanine, cytosine, or thymine. The sugar phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
Okay, so we all know that things can happen, mistakes can be made, but what does that mean if we have a base pairing mistake in our DNA molecule? Well, most of the time, mistakes are rare. So very rarely is a base placed in the wrong order because of their structure, because of the way that they chemically bind together, because we have one purine and one pyrimidine, and because we know we're always going to go A with T, C with G. But sometimes we do have a mistake, and those mistakes in base pairing in our DNA molecule are called mutations. So when you hear that word, it's just coming all the way down to the DNA level, and that means maybe we have an A paired with a G or a C paired with a T, and that's incorrect, and that can cause problems with the proteins that we're then going to make. We'll go a little more in depth as to the different types of mutations when we get into genetics, but just understand that a mutation in its most basic form is just pairing up of the wrong bases. Most mutations are not harmful to the organism, so a lot of your DNA does not actually code for protein, and sometimes changing one letter actually doesn't impact the structure or the protein made at all. There are some cases in which a mutation can be deadly or can cause a disease or disorder, and so some examples of diseases or disorders that are caused because of mutations are cystic fibrosis, sickle cell anemia, Tay-Sachs disease, and even color blindness. And we'll talk a little bit about each one of these as we move through this unit and our genetics unit. So before we complete our lesson for today, I want you to get some practice, and I want you to try and fill in the complementary side of this DNA strand. So I've given you one side of my DNA molecule. So our yellow little thin lines here are supposed to represent the sugar phosphate backbone, and inside of our ladder, our DNA ladder, or our double helix, are our bases. And so right now on this side of the molecule, you can see that I have an A, a T, an A, a C, a G, and so on. And so I want you to complete the other side using your base pairing rules. Once you've got that in your notes, make sure to double check them. I'll post the answer in just a second here, but I want to give you a little bit of time to try and figure that on your own. And remember your base pairing rules. So if you need to go back into your notes and see what those were, take a second to do that. Okay, so let's check to see if you got those right. So you should see that A's and T's are paired up. So if I know one side of the DNA molecule, it's really easy for me to figure out the other side because I know Chargaff's base pairing rules say that adenine always pairs up with thymine. So A and T, T and A and T. And then cytosine is always going to pair up with guanine. So if I have a C, I need to have a G. If I have a G, I need to have a C, and so on. And it looks like we do not have any mutations here, which is a wonderful thing. This is the end of the presentation. I will see you next time.